online. Welcome to the second episode of Attempting Science, the podcast. Once again, my name is Martin, and I will be the host for this episode. And with me, once again, we have
Emmanuel.
Welcome, Emmanuel. And if you are right now watching this podcast as a video, you're probably on YouTube because we are trying to mix things up here at this podcast. We'll try to
Be hip and integrate different
yeah,
medias.
so you can see our lovely faces. Exactly. And
Or at least Martin's face.
unfortunately, I hope you get my good side here. Anyway, but if you just want to listen to the audio file, then we are gonna still have it on SoundCloud for you all to listen. So we are just gonna. I think I'm just gonna start off with giving a shout out to iGEM Lund, and that because they at the previously weekend they organized the yearly this year's NIC, which is Nordic iGEM conference. And can you give a small description what that is?
It's like the mini iGEM meetup jamboree that is at Boston. So there's a posted presentation, a normal presentation, and also different social gatherings, a gala, and different stuff. And there are judges who determine who are the winner, and also for the judges prize, but also the iGEMers vote for the iGEMers prize.
Yeah, exactly. And so, I think we can kind of give a small summary how it went for us.
Yeah, we didn't win, but I think that we made a good effort, and we certainly weren't the worst. So I think it's a Swedish thing.
You can't say that on the podcast because that means someone else was worse than us.
No,
sorry if you're feeling. If you think we're pointing at you, we definitely not. I think we did. We could have done way better at. For example, I did the normal presentation, and some parts of my presentation I was pretty proud of, but there was a lot of stuff we could improve on. For example, we didn't have any statistics on on our project, and that I think that really just pulled us down into the.
And also, we didn't have a thing. All the others project has like Mars or cancer or something. We had different examples where our project could be used for. I think we should have one big thing, and also just mention the others really quickly.
Yeah,
we put too much effort on both things because we are doing our project for Alzheimer's and different neurodegenerative diseases, but also for industrial production. So I think if we just focus on one, we can do better.
So we were kind of like all over the place, unfortunately. Yeah. So that was a summary of our presentation. But otherwise, we loved it. We did really appreciate iGEM Lund's effort for
and also all the other teams that came with a great spirit, and
absolutely.
we met great people, and we had awesome judges that came with great ideas and great critiques.
Oh yeah, there was a lot of critique, but we do appreciate your feedback. That was we actually do. That was amazing. Thank you guys.
And also we had a great winner, Copenhagen University, who had a great project. So shout out to Copenhagen University.
Yeah, no,
they won both the Judge Award and also the iGEMers Award.
Yeah, for obvious reasons, really. They were really good. For example, what they were supposed to do with. We don't have to explain the whole project, but what they were supposed to do with this project to have it on Mars. And I mean that kind of gives the their ambition
Yeah, it is that
on
they're
on display.
Yeah, and they might change it later on, but it was how to make medicine that lasts longer, and for then example Mars. It was a really interesting and great presentation and project.
Yeah.
They had a little theater play and stuff, so it was really fun.
Yeah.
Yeah.
Yeah, absolutely. So once again, thank you so much, everyone who was participating in this competition, and I hope we'll see you in Boston.
So on to our next subject.
Yeah. I mean, so we have some couple of categories that we would like to discuss, I suppose. And I mean, Emmanuel, what was your main category that you wanted to bring up today?
Today I wanted to talk about organ growing and also both for humans and also for meat production.
Yeah.
So I think it's an interesting project because you combine genetics, you combine medicine, you combine so many different parts of science.
Exactly. So first of all, what is the problem? What is the problem that we're trying to solve?
The problem is that today there's a lot of people dying on the waiting list to get a new organ. So it's very different statistics, but around 20 to 35 or something % who die on the waiting list for a new organ. So there's very few organ donors. So by growing our own, we can solve this problem.
Yeah, exactly.
And also one problem is also meat production and how environmentally damaging it is. So if we can grow our own organs and also our own meat, that could be great.
Yeah, exactly. And obviously that has a lot of more that can also solve a lot of ethical dilemmas that we have in the meat production. Obviously, because I mean, well, there's going to be still some ethical dilemmas with this kind of technique, but that will at least solve the inhumane way what do you say?
Slaughter the animals,
Yeah,
and
I
also
suppose.
how we treat them.
How we treat. Yeah, that was the thing I was supposed to say. Yeah. So yeah, but okay. So there's a lot of ways how you can produce these organs, and first of all, I mean, we have two different examples that we would like to discuss today, and that is the first one that we create these chimeric cells or chimeric organs, which we will later on describe what that is and so on. But the other one is
growing the own with your own cells. So one is growing the organs in a human or animal host, and the other is growing the organ out of a body in a lab.
Yeah, exactly. So the chimeric cells that I previously mentioned is that we take cells from the human body and then implement them or inject them into a pig embryo, for example.
Yeah, and then put it in a surrogate pig.
Yeah.
To later grow. So should we go into the mechanism behind it?
Absolutely, we can do that.
I think that everyone are curious.
Okay, so well, it's real hard to start off with the most basic, but let's start with that. So what in our bodies we have cells called pluripotent stem cells, and what these stem cells do and what's so special about them is they can differentiate into other types of cells. For example, they can they can become skin cells. They can become heart cells, muscle cells.
Yeah, pluri stands for like several potent for different. How should I describe different?
I have no idea.
Yeah, different ways to become.
Yeah. Exactly, and for example, so maybe you're wondering right now, like, oh, okay, so wait, so skin cells can become like other types of cells? No, they cannot. If a skin cell have become a skin cell, they can turn into another kind of cell.
Yeah, it's
basically.
like getting on an airplane and then wants to go back, you know,
Yeah.
or go to a different destination. No, you're on that plane, you can't go anywhere.
Exactly. So, exactly. But the stem cells, they can become any kind of type of cell. So, and researchers have used these stem cells by injecting them into let's take pigs as an example, because we use pigs in order to produce human organs, because we have kind of same temperature in a body.
Kind of same, like size of the organs.
Exactly. So they're very good hosts for us, surrogates for us. So what they do is they take human stem cells and inject them into a pig embryo. And what they do also with this pig embryo is they genetically modify it so it can't produce a certain organ. What they can do, they can kind of make a scaffold of the organ. They can help they can help stem cells to grow into this organ, but the pig's own cells cannot produce this organ. It can only help the human stem cells to become the organ that we want. So basically, the only types of cells in this now pig embryo who can produce, for example, let's say, what is good? Pancreas, perhaps?
Yeah, or thyroid or something.
Yeah, let's say, let's say a pancreas, for example. So the only type of cell in the pig embryo who can make this, who can produce, can differentiate into a pancreas is our human stem cells. So it cannot naturally just they naturally just grow into this pancreas, and yeah.
So there aren't any competitions between the pig cells or the human cells, and also it limits the graft versus host disease, because if you have a pig heart or something or a pig pancreas in you, the body will say, yeah, that's not your cells, they shouldn't belong here, and they will be, the immune system will react. So by eliminating the pig cells, we can just have the human cells, and it's really great.
Yeah, it's when you, I think you went a little faster.
Yeah.
I think what you were trying to say is when we are unfortunately killing the pig in order to get this organ, so a human can get this organ, it's easier for the human body to accept this organ, because obviously if it wasn't a problem to implement another animal's organs into a human body, we didn't need to make all this effort into making these chimeric cells in the pig. So for example, we don't just take a regular pancreas from a pig and just not transport, but transplant into a human because, as you said,
Yeah, the cells has different proteins or different like flags that say where they come from, and these are different for different kind of individuals and also different kind of animals.
Yeah.
And the immune system would react on these flags.
yeah.
So-
So exactly. So our immune system will like, okay, this is not part of our body. It has to go.
Yeah.
So it starts to attacking the
Exactly.
organ.
And one of the first examples of a human or an animal organ in a human was ended horribly because it wasn't a human organ, so it just rejected it. So that's why it's needed.
Yeah, absolutely. So yeah, that was a basic of how you make these chimeric cells, and I guess we can also explain what chimeric cells is. It's that it describes two different types of cells with it's two cells who have different genomes in them. Yeah, exactly. I think you can say that.
And that's one of the methods, but there's another you mentioned. What was that?
Did I?
Yeah.
You did.
I did.
Well, it's that we well, I think you can explain as 3D printing this organ or own organ without a host. Yeah, no. I have to admit I don't know a lot about it. Because I've been having a question like, okay, how do they make it? Like what is the, for example, they use biomaterial, but what is that? Is that like just take a could you explain that for me?
I'm not an expert on 3D printing, but I think that the 3D printing is first just to make a scaffold by maybe collagen or something, and then the printing can insert human cells in that. On that they can grow. But there's also one method that's a bit more inhumane, where you take animal organ and then wash it clean of the animal's cell. And then the only things left are just a white scaffold of, for example, a heart. And then you can start to grow human cells on this scaffold. So I read an interesting similarity between, taking an apartment and just take away all the decorations and the sofas and everything, and just recreating the apartment, just putting in a new decorations and stuff.
Yeah.
So it's really like renovating a heart, for example, or a different organ. It's
good way to describe it. Yeah. Yeah. I mean, so when you hear all these techniques, what do you think about the ethical dilemmas with it?
I think one of the dilemmas are the animals where, for example, if we should talk about the chimeric organs,
Yeah, go ahead.
You need
Choose.
to have an animal host. And first you need to gene edit this embryo. And then you need to put in the human stem cells, pluripotent cells. And then you need to put this embryo into a surrogate. And then you need to kill the pig after when it's born. It has grown a bit to take out the organ, and that isn't very nice, I think.
Yeah,
And also, when you put in human cells, there is a possibility that human cells can take a different route in the embryo and help with other organs or interact with the pig cells in a different way.
Yeah, I mean, if we compare it to the regular arguments with meat production, or like when we breed pigs and other animals for food, one of the arguments that meat eater use is, well, at least animals have a decent life before they die. And okay, sure. I mean, according to me, I'm a meat eater, so I'm kind of buying that one argument for now at least. But when we're talking about using pigs as almost like a just a host for,
you can't use the pig afterwards. I think that most people won't want it for meat or something.
Yeah.
Because what if there's a human cells in the meat?
God, yeah.
You
Oh,
can
yeah.
become like a cannibal or something.
No,
or maybe
exactly.
interact with the pig brain and sorry.
No. Because, that was a real good point, because there is a lot of, because unfortunately there isn't 100% accurate that the stem cells from humans will only become this organ. Unfortunately, not a lot of times, but sometimes these stem cells kind of I don't know if you,
They can interact and
escape from the organ that we wanted to produce, and kind of move on to other parts of the pig, for example, the brain, and thereon they can differentiate into a part of the pig brain. And
for example, the neocortex that make the pig smarter. The neocortex is the outer part of the brain that is correlated between intelligence.
Exactly. So and at that point we're kind of making a new breed. I mean, we're making something like a mix of a pig and a human.
There might be some research how to limit that.
Yeah.
For example, to put a kill switch, a gene sequence in the cell, and then activate it and kill the cell. But it's really difficult to detect it, and there's so many different situations.
Yeah.
So maybe researchers don't want to risk it. Who knows?
Maybe. But if we go back again to the argument that meat eater use, that the animals at least have a decent life before they die. Well, I don't know how long. If we go back to the pigs that we use to produce organs, how maybe they? I have no idea. Maybe they have a pretty decent life before they die.
They need to have at least a life where they can't interact with too much bacteria. So maybe they're inside a lot. I don't know.
Yeah. I mean, from a pragmatic point of view, it could kind of be the same. I mean, if for example, let's say that the pigs that we are using for organ transplants or organ hosts, they're the host for our organs. If they have a decent life until they're killed, I mean, then it is on the same level as the meat production.
Yeah.
But if then we can go into the aspect of changing a living organism's or a living animal's own genome. I mean, how do we? How are we sure that they won't be in constant pain from this genetic modification that we did in the beginning?
And also, often it seems to often the researchers use CRISPR-Cas9, and it's a really good gene editing. It's one of the best, but there is a chance of it doesn't cut in the right place or put a mutation somewhere and can mutate the pig in some ways. So a different organ or like a mutation somewhere that can harm the pig. And also maybe that mutation can get into the organ, and then the human who gets the organ gets the mutation and doesn't have a decent life. Maybe the organ fails or something.
Yeah.
So there is so many factors that play a role in this evaluation.
Yeah, exactly. I mean, yeah.
And also, for example, if if you can grow organs, you can grow meat, which is great to decrease the meat consumption. So that would be a great way to go. But the problem there in the meat, if you want to grow your own meat, is that you use, when you grow cells, they need to have a nutrition or some kind of food. And that often is from cows. When a cow is slaughtered, you can, if it's pregnant, you can take the uterus and take out a medium or the food for the cells from there. So if you grow the meat, you still need that animals are slaughtered. So it's a really difficult dilemma. But maybe in the future you can grow a different medium or food for the cells.
Yeah, exactly. I mean, maybe we will be able to make mediums from plants instead. That will be able to.
Or maybe we can grow our own mediums. But before we rehearse always a bit before the show. And one example you gave that was quite interesting was animal diseases getting into humans.
Oh yeah, I mean, as most of us know that that other animals are more prone to get other diseases than we humans are, and that is because and these diseases, well, these bacteria or viruses can make use of a certain molecular pathway, a molecular mechanism in cows, for example, or in animals that does not exist in humans. So they cannot infiltrate, they cannot infect us, but they can only affect those organisms or those animals that have this molecular
likeness or a molecular pathway.
Pathway. I mean, you probably don't know what molecular pathway is, but that's what
it's a chain reaction of chemical reactions that results in different things. You know, for example, the cell dividing, it needs a molecule that sets off a chain reaction.
Exactly.
And if you have animal cells in you, from example, the transplanted organ, you can get the disease, and maybe the disease can start to integrate with human cells and start to make a disease that are also able to infect humans.
Yeah, exactly. I mean, yeah, absolutely. And I just previously read a lot about it and on how, for example, retroviruses can integrate into animals' genome and
retroviruses are able to, override or rewrite the DNA. For example, HIV is a retrovirus.
Yeah.
So yeah, it's
exactly. And for example, so if we take an animal as a host for our organ production, who has been affected or infected by this retrovirus, there has been this overwrite from our retrovirus. We can then later on we will have this retrovirus sequence in our own bodies, which can cause a lot of problems, I suppose we can say that. I mean, just we have no answers for this whatsoever. We have no answer for the ethical dilemmas, obviously. Otherwise, we wouldn't be here. So anyway, that was about production of organs. So that was I mean, I would love for us to for another episode of a podcast, we can actually get into the meaty and gritty of this of these techniques and methods. I mean, I think we only touched upon like the surface of this subject, but we can move on to our next subject, which is
An update in our projects.
Oh yeah, we should do that as well, I suppose. Yeah,
And maybe then end this, I think. Then.
Okay, yeah, we can absolutely do that.
I'm too tired.
I thought we talked. Anyway, we can do an update and then just say bye, I suppose. So, all right, updates. What are we doing? That is actually a question. I have no idea what we're doing.
Yesterday we started at the lab, so we got an introduction and started to make different preparations because, for example, when we grow our bacteria, it will need different stuff. For example, nutrients and stuff like that. So we're preparing that. Me and Martin made agar, which is a medium for the bacteria to grow on.
Yeah.
And also a part of iGEM is to try out different iGEM teams biobricks, different gene sequences that makes different stuff. So we're taking a look at that and what biobricks we can use.
Yeah, exactly. And that will be a good introduction for those in our team who haven't been working a lot with, gene editing. I suppose you can say that. For example, I was previously in Denmark at the DTU. It's a university there. I don't really know what DTU stands for, but I suppose
Denmark
university.
Technical University.
Oh, there we go. Perfect. I suppose I need to look at you more than the camera. I'm like, hey.
I feel a bit lonely.
No, I'm sorry. Anyway, yeah, exactly. And at Denmark's Technical University, we're working with some biobricks. It was like a small introduction on how are you going to, take these sequences into the bacteria, so the bacteria can express that, it can use these sequences to make the protein that you want.
Yeah, for example, just a fluorescence that makes.
What is that? Fluorescence.
Fluorescence is just light emitment, so the bacteria will shine in a certain color.
Yeah, exactly. So if you take this biobrick and you kind of inject, so to speak, inject into the bacteria. You don't actually inject in that kind of way, but it's pretty much the base. That's kind of the basic of it. That's a way you can describe it. So you inject it, and then the bacteria can use this sequence in order to make this kind of light, and you can see then your bacteria will change color into, either green is often the color you use, but you can also use red lights and a blue light and all kinds of lights, I suppose. So we're doing that. We're just doing a small introduction, and then I'll do the podcast. Will be a lot more podcasts coming up soon. We won't give any spoilers of what they will be about, but just stay tuned. Anything else? It's a small. It's the first week of the real work, I suppose. So it's we have started to trying to get into it, and.
We still can't find everything in the lab. We just ran around to find like tape and stuff. So
that was awful. Our project leader just looked at us as mice in a small. I don't know what you can say. Like lab rats, just running around. Didn't know. Like where places were, and just
because
screamed in panic. Where's the protocols? No, not protocols. That's That's not how you protocols. Where's the protocols? That's what we panickedly screamed. So should we just end this now? I suppose. All right. That was the second episode of the Attempting Science, the podcast, and and we will just end this so we don't screw this up anymore. Bye.
Bye.